Good afternoon, everyone, and welcome to the One Health Seminar Series. The series is organized by the University of Guelph One Health Institute and graduate students enrolled in the One Health Collaborative Specialization. My name is Jamie Galtz, and I am an MSc student in the Department of Population Medicine and also one of the One Health students. It is my pleasure to be facilitating this seminar today. With great respect, we acknowledge that the University of Guelph resides on the ancestral lands of the Attawandaron people and the treaty lands and territory of the Mississaugas of the Credit. We uphold the significance of the dish with one spoon covenant and the continuing relationship our Indigenous neighbors have with this land. We recognize that this is a gathering place and that all places we're joining from today is today home to many First Nations, Métis, and Inuit people, and acknowledging them reminds us of our relationship with this land where we explore, share, discuss, and learn our shared responsibility for human, animal, and environmental health. Before I introduce our speaker today, I wish to inform you that we are recording this seminar. The recording will be posted on the One Health YouTube channel, and a link will be made available on the One Health Institute website. The recording may also be used by the One Health Institute in the context of promoting and showcasing One Health work being done on our campus. If you have any questions or concerns about the recording or One Health generally, please contact us at onehealth@uofguelph.ca. In addition, we have enabled live capture for this presentation. If you wish to see the captions, please click on the settings option at the bottom right of your screen and check the English option. At the end of this seminar presentation, you will be able to ask questions. As this is a Microsoft Teams live event, you will need to ask your questions through the chat function. To open the chat function, click on the Q icon on the top right of your screen. You will see the featured questions. If you wish to ask a question, click on My Questions and type your question in the box at the bottom of the screen. To ensure that everyone is familiar with the chat function and to help us know who is joining the seminar, please take a moment to indicate if you are an undergraduate student, graduate student, or faculty or staff and from which department you are from. If you are joining us from outside the University of Guelph, please tell us which organization you are from. Wonderful. Thank you for sharing. It is now my pleasure to introduce our speaker for today. Dr. Hanner is a professor in the Department of Integrative Biology here at the University of Guelph. His lab studies molecular biodiversity in both aquatic and terrestrial environments, with an emphasis on developing DNA-based tools for biomonitoring. He collaborates closely with the Canadian Food Inspection Agency and other partners on the development and application of these tools to support animal and plant health, food security, and environmental monitoring objectives. Today, he will be speaking about the role of DNA barcoding and environmental DNA-based biomonitoring in support of a One Health agenda.
Good afternoon. Can you see my slides?
Yes, we can. They look great.
All right, let's move into presentation mode then. I'd like to thank the organizers for inviting me to participate in your One Health seminar series. It's an exciting opportunity for me to showcase a bit of what we're doing here at the University of Guelph in terms of molecular biodiversity research and how it can feed into a One Health agenda. So to preface the DNA barcoding aspect of the talk, I really like this quote from Bob May. It was in an article in Nature titled "Taxonomy is Destiny," where he said, "Without taxonomy to give shape to the bricks and systematics to tell us how to put them together, the house of biological science would be a meaningless jumble." So in terms of trying to address taxonomy, this issue of naming species, We owe our current framework of using Latin binomial nomenclature to Carl Linnaeus, where we began to develop a lingua franca for science, where we could get away from colloquial names that might be applied very differently in different settings to using scientific names to communicate about biodiversity. Now, of course, during Linnaeus's time, this was seen as a bit like stamp collecting. Species were largely thought to be immutable creations of God. So it wasn't until about 100 years later, with Charles Darwin's thesis of descent with modification by natural selection, that we began to understand the mechanism for the origin of species. In other words, species exist because they evolve. And some species are more closely related to each other than others, which can often mean telling them apart is difficult. So as we try to understand the biological world around us, we use this infrastructure of taxonomy that involves collections of specimens in musea and herbaria. They're described according to taxonomic codes of nomenclature. We have different compilations of what are thought to be valid taxonomic names. Quite often, our species concepts need to be revised. Some names are considered no longer valid as they become synonymized through taxonomic revisions. We're working with large data repositories today. We have to consult monographs. And largely, all of this information is undigitized, so it can be very difficult to access. Add to that the fact that most of what we know about taxonomy stems from adult, intact specimens, means that it can be very difficult to extend our knowledge of known species to juvenile life stages or processed products. So even the species that we have names for, we're challenged to apply this taxonomy when we're dealing with damaged specimens that we would like to identify or early life stages. So it's important also to recognize that the museum specimens that we've built our taxonomic backbone on are really looking at phenotypic variation, and that can be influenced by the environment. So trying to circumscribe what a species is based on looking at a handful of specimens in a given collection can be challenging because it's hard to differentiate what are traits that can vary within a population and perhaps even through environmental influences versus characters that are fixed morphological differences between species. So really the challenge for us with respect to biodiversity, it's a key measure of ecosystem health. But frankly, because most of our taxonomic information is undigitized, we don't really know what we know about biodiversity. It's difficult to measure changes through time, and this in turn makes it difficult for us to determine whether our resource use decisions around land management or other anthropogenic activities are sustainable or not, because we have this biodiversity gap in our biodiversity knowledge that somewhat stems from a taxonomic impediment, that we just don't have enough taxonomists today working on describing species to scale this up. So we talked about the known unknowns of taxonomy in terms of all of this stuff existing largely in an undigitized framework that can be difficult to access. And what we think we know about biodiversity is that there have been about 1.7 million of species of plants and animals described using this Linnaean taxonomic framework, the vast majority of which are insects. If you look at this graph on the left, each one of these squares represents 10,000 species. So then the challenge is actually extrapolating to answer the question, how many species are on Earth? And we don't even know to an order of magnitude how many species are on Earth. Using data from multicellular plants and animals, people have extrapolated that there could be as many as 10 million species on Earth, but other folks studying protists have said there are probably 20 to 50 million species of protists alone. So there could be as many as 100 million species on Earth. So the reality is that we know very little about the biodiversity around us in terms of total biodiversity. So I talked about our taxon concepts largely being based on phenotypic variation assessed looking at museum and herbarium samples. But now fast forward into the 21st century, seeing the writing on the wall with DNA sequencing, we can now directly interpret genotypic variation. And it's the future of taxonomy really is through integrating our traditional knowledge with DNA sequence-based approaches. This is really what's going to allow us to scale up. Now, in many cases where we don't even have a valid name or a species description for some taxa, we may only know them by a sequence that differs from everything else in our libraries. And I'll get to that in a moment. So first I want to talk about genomic technology. The human genome was sequenced at the end of the 20th century. It was really an expensive endeavor at the time. And what we were looking at there was sequencing all of the genes in a single species, more than 3 billion base pairs. Not a trivial undertaking. What I'm going to talk about today with respect to DNA barcoding is more of a horizontal genomic approach, where we're trying to sequence one or a few marker genes across all species. So while the sequencing requirement for any given specimen is much lower, the challenge of developing a pipeline to get access to expert identified reference material to begin to sequence biodiversity is non-trivial as we try to source materials from all around the globe. So I want to get to this new tool for taxonomy, this idea of DNA barcoding that was first introduced by my colleague Paul Hébert here at the University of Guelph, where he began to recognize that this ability to compare genotype information across a huge range of organisms is a very powerful tool. So what is DNA barcoding? It's a method of species identification based on DNA sequences derived from standard marker genes. So in animals, we use a mitochondrial cytochrome oxidase 1 gene but however, the mitochondria doesn't evolve fast enough in plants to be useful as a marker. So we're looking at chloroplast genes, RBCL and MAPK as our barcode markers for plants. And then in fungi, we're using an energy spacer. So we do have to pick markers that will discriminate species in different taxa. There isn't one universal barcode that we can apply across all of life. But the hypothesis is that for that gene segment, each species will have a unique sequence, and that sequence is termed a barcode. So you can see in the little cartoon example at the bottom, we have a short fragment of DNA sequence. DNA is, of course, made up of an alphabet of just 4 letters representing the 4 bases of which it's composed. And over time, random genetic mutations will start to creep into the genome of different species. And we can use these mutations to tell species apart. So you'll see species B has a couple of nucleotide base differences from species A. And it's these combinatorial differences in their nucleotide sequence of the barcode gene that allow us to uniquely identify species. So some of the necessary characteristics of a target for DNA barcoding require that there's very little variation in a species, but high variation between them. So in other words, there's this barcoding gap that we can use to tell species apart. This barcode region has to be flanked by highly conserved regions so that we can make near universal primers that will amplify, say, all plants for one of these target markers, making it easy to amplify. And then ideally, to keep this a rapid and cost-effective tool, we want to use short enough barcode fragments to have enough information to tell species apart, but still only require a single Sanger sequence run to generate that barcode to help keep the cost down. This is the work that is done at the Biodiversity Institute of Ontario and Center for Biodiversity Genomics here on the University of Guelph's campus. It's the global headquarters for the International Barcode of Life project, which aims to build a DNA-based library for all species on Earth. The workflows are fairly straightforward, using specimens that have been identified by experts. We take a tissue sample, extract the DNA, PCR amplify the barcode, and sequence it. We put that into the Barcoding of Life database along with information about who collected the specimen, where it was collected, the GPS coordinates, digital images of it, so that we have a lookup table that includes both species names and barcode sequences that we can use as a basis of identifying an unknown. By comparing a sequence to our reference library, we can often match it and use that to identify a species. So barcodes, again, are trying to develop a reference library for known species. It'll give us this master key that can allow us to ID all life stages, give us IDs in a fast and inexpensive manner with relatively low taxonomic uncertainty. So some folks have looked into this idea of error rates, how well barcoding works. If you look at the graph on the right, you'll see this kind of barcode gap when variation within a species is low and there's a lot of divergence between its nearest neighbor. We can easily tell species apart using barcodes. In some cases, there may be overlap where the within species variation and the between species variation aren't clear and distinct. That can arise for a number of reasons, maybe because our traditional species concepts are simply wrong. What we think are two different species may only be one. Or in other cases, there may be a large amount of standing genetic variation within a named species across its range. So the point is, barcoding isn't perfect, but it is very good. If you look at this graph of several hundred species of marine animals that the authors Meyer and Pauly studied in this paper, you'll see that for the vast majority of species analyzed, the distance to its nearest neighbor is greater than the distance among populations of that species. So in other words, for all of the species across the x-axis, where the orange dot is above the black line, that suggests barcoding works very well. You'll see a few cases here where this interspecific and intraspecific divergence overlap. So in some cases, we may only be able to identify a specimen to a pair of closely related species. But typically, at least in animals, for the mitochondrial cytochrome oxidase 1 gene, we have very good discriminatory power among animal species with the barcode. That said, we have to differentiate between DNA-based identification and DNA taxonomy. So when we have very robust taxon concepts for species that have been well studied, like fishes and birds and many insects, we can use the DNA database to identify unknowns. And we have very little ambiguity about the identity of that species. But in some cases, when we're looking at soil invertebrates and other groups that have very little morphologic character variation to allow us to write traditional species descriptions, we just don't have very good knowledge of the species diversity there. And so in this range, we may only know a taxon from its DNA sequence. There may not be a name to attach with it. As I said, there are probably 10 to 100 million species. We've only formally described about 2 million, and of those, we've only been able to generate barcode data for about 600,000. So we're making rapid progress, but you can well imagine there are many species that don't yet have names or barcodes. So in these cases, when we're doing sequencing, we simply know them by a sequence. And in this case, that's essentially DNA taxonomy. We can't identify something to a named species because it just simply hasn't been described yet. So there's a lot of pedagogical value in barcoding in terms of integrating fundamental concepts of evolution, phylogenetic theory. We marry that with practical methodologies like PCR and DNA sequencing, phylogenetic tree reconstruction, and all of this has a basic but far-reaching application in terms of being able to identify species in a variety of applied contexts. So we can use barcoding as a research tool for improving our species-level taxonomy, associating different life stages to a single species that may have previously been thought to be two different species. There are many things where there's sexual dimorphism, the males and females don't look the same. So without seeing them in copulo, you might conclude they were actually different species. We also use it as an applied tool for identifying things like disease vectors, environmental indicators, at-risk species. And then, as I mentioned in that sort of example of DNA taxonomy, we use barcoding as a triage tool to alert us to the fact that there are new sequences that don't match anything. This is something that we need to pay attention to, and ideally through integrative taxonomy, describe that species and give it a name. So there are a lot of benefits from barcoding. We can standardize the application of names and facilitate IDs. We can highlight specimens that represent a range expansion of a known species, flag this kind of unrecognized or cryptic diversity, and really demonstrate the value of taxonomy to society. So that's why there's so much interest in barcoding for looking at things like regulated pests, disease vectors, plant pests, and other things. So one issue directly impacting human health are bird strikes. This is thought to cost the aviation industry on the order of about $400 million a year when a bird gets sucked through the turbine of the engine of an airplane, and all that's left is a bit of spatter on the fuselage. Pretty hard to ID what bird actually hit the plane. But if we can ID the species and know something about the life history of that species in terms of when it migrates or whether it's flying at a low elevation, we can start to try and figure out ways to minimize our overlap of flight paths with migratory paths of birds and minimize these kinds of airstrikes. We can also use it to support human health by looking for evidence of ruminant waste in cattle feed that might transmit bovine spongiform encephalopathy or mad cow disease. So being able to take something like a feed product and detect the presence of bovine DNA in it is a powerful tool to help make sure that we don't see further outbreaks of mad cow. Obviously, international trade, we're in a globalized society. Materials are moving all around the world, and this leads to a number of biological invasions. We would like to be able to have early warning signs. If we could detect an invader before it was well established, we would have a reasonable chance of eradicating it. But right now, we don't have, or we're just now starting to develop approaches that can allow us to have these kinds of early detections of invasive alien species. And that's really important because the cost of these invaders is non-trivial. The zebra mussel is thought to cost about $100 million a year in terms of having to clean out water intake pipes and other things that they clog, not to mention some of the environmental concerns that they bring in terms of displacing native species. The emerald ash borer and the $500 million club. The pine beetle is thought to cost the Canadian economy somewhere in the range of $1.5 billion over 10 years. These are all invasive species that perhaps could have been caught early and eradicated before they became established. So we really need to think today in the Anthropocene about environmental degradation and how to mitigate some of its impacts, how to restore environments once they've been degraded. The bottom line is that species ID is crucial in all of these endeavors. It's important to many, many different aspects of our One Health agenda. The whole point is that if I can get a sequence and compare it to a database, then I can match that sequence into the Barcode of Life database and get a name. Once you've got a name, you can Google it and find out all sorts of interesting information of where it lives, whether or not it's edible, what its conservation status is. So that ability to put a name on a specimen is really crucial. And I want to talk about a few examples in our lab where we've used DNA barcoding. In this case, we were trying to understand food webs and trophic interactions between our native top predators like lake trout versus lakes that have seen introductions of invasive species like smallmouth bass. So there have been some visual observations about what they eat, not terribly conclusive, but it was thought that basically your lake trout that were pelagic feeders weren't really overlapping with the smallmouth bass that are nearshore feeders. And so we thought to test that by collecting the stomach contents of fish from a variety of boreal shield lakes in Canada and then barcoding these individual bits in the stomachs of the different species for which we had collected stomach contents. And you can see from the pictures, you know, at the top left, this small piece of vertebral column would be impossible morphologically to identify the species. But in this case, we could ID it to yellow perch, bits of frog. We were able to ID to species using barcoding. And even if you look at the bottom, we were also found tapeworms in some of the stomachs, and we could identify those to species as well. So barcoding was a pretty interesting tool in terms of allowing us to address this question of habitat coupling and potential competition between native species and the invasive smallmouth bass. And the traditional view was that there was very little overlap, but as we began to barcode them, what we saw was that the open water species were feeding on many of the same things as the bass at the near shore and vice versa. So there is a lot more movement in these systems than was previously thought, and probably a significant amount of competition with the invasive species as well. Just to give you another interesting example of how we've been using barcoding in the lab, this is Diagnostics gigas, or the Diablo squid, traditionally found around the Gulf of, or the Baja California. It's a 2-meter long, pretty voracious predator of fish. And with climate change, its range has been moving northward up the west coast of North America, recently arriving in British Columbia. And fishermen were worried, is this feeding on native species that are of commercial value to us? Why are these things washing up dead on our beaches when they were thought to be more of a warm water species? What's going on here? So one of the students in my lab, Heather Braid, went out to the west coast, collected some of these dead squid that had washed up on the beach, brought them back to the lab to dissect out the stomachs, barcode the stomach contents, and we got a list of a number of different species, many of which are commercially harvested. And some of those actually bioaccumulate paralytic shellfish toxin, which we also detected in the bodies of the squid, which suggests maybe it's the paralytic shellfish toxin that's killing them and causing them to wash up on the beach. Now, that may have implications for other scavengers, like the black bears that are eating them. We don't know to what extent the toxicity from these squid may impact the bears. And interesting to note, we're now also seeing this Diagnostics gigas or Diablo squid showing up in the human food chain. So, you know, enterprising commercial fisheries are harvesting this animal, and it does represent a potential health threat that could magnify up through the food chain. And I want to focus on that for just a minute, because as we've built DNA barcode libraries for fishes of the world, one of the students in my lab was curious to know, is our reference library mature enough that if we bought fillets from the market, we could barcode them and match them to the library? Now, this was over 10 years ago when we were just getting started with the Barcode of Life initiative. And even then, we had enough data in our database for commercially important fishes that we could ID all of the fillets that we encountered in the market. But what we were surprised to find is that 25% of them were mislabeled, always a species of a lower economic value being substituted for one of a higher value. So this economically motivated adulteration is what we would call food fraud. This is a problem that we've seen not only here in Canada, but around the world. It seems to be systemic within the industry globally, in part because once you fillet a specimen and all the morphological characters are removed from it, it's very, very difficult to ID what species it is. So it's an easy target for fraudsters. Now, one of the things that we found that was a bit disconcerting is that escalar, or oilfish, is often sold as white tuna or swordfish or a variety of other species, but it has a waxy ester in its flesh that about half the human population lacks an enzyme to be able to digest, meaning that if you eat more than a couple of ounces of its flesh, you're going to experience stomach cramps, a nice explosive orange diarrhea, not the kind of thing that you think you expect to get when you're paying for your sushi dinner. So while carriuria isn't fatal, it's not a pleasant experience. And I can tell you that we have found escalar being served at sushi restaurants right here in Guelph. It gets a bit more nefarious than that, though. We were asked to collaborate with the U.S. Food and Drug Administration when the Chicago Public Health Authority recovered a meal remnant from a couple's stove. They had bought what they thought was a species of a commercially available species of fish, monkfish, but then they ended up in the hospital with severe poisoning, and they couldn't ID the species of fish in the soup. So they sent it to us for barcoding, and we actually found that it was a pufferfish. And FDA was very skeptical about that because that's a highly regulated species because of its known toxicity. So they did a toxicity screen and found tetrodotoxin, the toxin made by pufferfish. So we had two independent lines of evidence that pufferfish were getting into the food supply and poisoning people. Unfortunately, that was not a one-off incident. CDC has reported a number of these kinds of outbreaks using DNA barcoding to identify pufferfish. So these kinds of, I don't think that this was an economically motivated fraud issue. I just think sometimes pufferfish look similar to other species and may get into the food chain as a contaminant, but the point is it represents a significant public health threat. It also, this kind of mislabeling is an impediment to consumer choice. And what we're seeing is that overfished species that are either illegal to harvest in a given area or at a given time can be laundered into the food supply by mislabeling them as something else. So it has sustainability implications for managing our wild fish stocks as well. And this isn't just seafood. We've seen this happening in herbal teas as well. You know, if you've ever wondered why herbal tea tastes like straw, it's because it's found in about a third of the tea bags that were tested in this study, but it's never on the list of ingredients. You know, straw is just a cheap filler that they can put in there to bulk up the tea bag. So again, fraudulent dilution kind of practice. Similarly, my colleague Steve Newmaster and folks in his lab here at the University of Guelph decided to start looking at natural health products and herbal supplements and found a significant amount of adulteration and mislabeling in those products as well. So we need to use these barcoding-based tools for surveillance of our food supply for a variety of reasons, for human health, for environmental health, and also more broadly, food security. We've been working with the Agriculture and Agri-Foods Canada and Canadian Food Inspection Agency to develop a barcode library for weeds that are invasive and can compete with our crops so that we can use this to look for seed lot contamination and help identify the weed bank in soils. Lots of applications. This study I thought was interesting because folks looked at ferns being sold in the United States that were marketed as a North American species, and they found that nearly half of them were incorrectly identified and that they were actually international species that were being brought into North America, sold as a North American species, but were not in fact the species. Well, why does that matter? Because live plant imports are the major pathway for forest insects and plant pathogens to get into North America. So this lack of quality control or perhaps even fraudulent movement and sale of plants opens up pathways for disease transmission and invasive alien species of plants and also animals. So one of the things that my lab was interested in was being able to use these kinds of tools to identify arthropod pests for plants. We started to look at a variety of regulatory programs from Canadian Food Inspection Agency through to the Europeans. To our surprise, there was no single list of quarantine plant pests. So our first job was to coalesce all of these different lists that were regionally focused into one master list, query that against the barcode reference library, so that we finally had a master list and could do the gap analysis. And we're seeing a lot of activity in terms of barcoding reference specimens for plant pests of quarantine significance, which is good. That means we can start using this as a tool to identify these plant pests. However, we sought to do the same thing for parasites and vectors of zoonotic disease. Once again, there was no comprehensive list. We spent a lot of time trying to pull lists from different sources to build up our list of names for which we could query the barcode library, do this gap analysis. And we were a bit surprised because the parasite community was one of the early vocal proponents of saying barcoding could be a big help in identifying parasites and vectors of zoonotic disease. But what we found was that after we constructed the checklist and queried it against BOLD, was that fewer than half of the species on the list actually even had a single barcode in the library. So there's very little data, or in other words, we have a long way to go to building these barcode libraries for parasites and vectors of zoonotic disease. What you can see on the figure on the right is the yellow bars represent the species in these different groups of ectoparasites, vectors, and hazards. We broke them down into different functional groups, and you can see the yellow bars represent species for which there's no barcode record. We haven't even begun to characterize these species genetically. So if we want to use a tool like DNA barcoding to identify parasites and vectors, we've got to do more work in building those barcode libraries. And this is important because human zoonoses account for 61% of all infectious disease agents and 75% of all emerging diseases. Now you think about that example with plants moving pests around in international trade. We're seeing approximately 500 million live vertebrates being moved around in international commerce every year. So this potential for movement of parasites and vectors is significant, and it represents a real challenge for One Health. We've done a bit of work in the lab with DNA barcoding ticks to try and build a barcode reference library for them. And as we've extracted the DNA and sequenced it to generate a barcode for that tick, we also used a PCR test for Borrelia burgdorferi, the causative agent of Lyme disease, and could see that in some cases, even from a dead tick or a shed exoskeleton, we could demonstrate that they were competent vectors of Lyme. So we can kind of do double duty here by building our barcode libraries and also querying for different diseases of interest. And in this case, the barcodes matched the known taxonomy pretty well, suggesting that, you know, barcoding is a good tool for identifying ticks. And this gets me back to this idea of the Borrelia burgdorferi test, or using qPCR for rapid molecular diagnostics. The beauty of DNA barcoding as you sequence an unknown is that you can compare that sequence to a reference library and identify it. But that has to be done in a laboratory. For many applications, we'd like to be able to take these kinds of diagnostic tests to the field. So using this multiple sequence alignment of all of these barcodes from all of these species, we can exploit the species-specific patterns of genetic variation in the barcode to make species-specific primers and probes for a qPCR assay that will allow us to confirm the presence of a particular agent. So something like Lyme disease, or in this case, we've developed qPCR tests to identify the various salmon species that actually can now be deployed in handheld instruments. This is one of the exciting things, is that we're moving qPCR out of the lab and into the field, where you can do DNA extraction and PCR on site in real time to get an answer if there's Lyme disease in a particular tick, or if this fillet is really coho salmon. And we've developed some of these kinds of qPCR assays to differentiate aphids that are quarantine pests, in this case, Aphis pomai and Aphis spiricola. they look alike. To be able to identify them morphologically, you'd have to send them into a taxonomist who would clear and stain and slide mount the specimen, study very fine-scaled structures to identify them. So if you're bringing in a truckload of, say, cut flowers into Canada, and they suspect you might have one of these quarantine pests, you're going to be waiting at the border for a couple of weeks to get a classical morphological ID, which is essentially tantamount to rejection of your load. It's going to end up in a landfill because it spoils. So we see real value in bringing some of these kinds of tests right to the border where people can use them. Now it's time to shift gears and think a bit about species scans. We can use high throughput sequencing to do massively parallel barcoding on mixtures. So as we build the barcode reference library, we're using Sanger sequencing, which generates a single sequence from a particular specimen. We use that to build the reference library. But as we start to want to look at mixtures in the real world, we can use this high throughput sequencing to generate barcodes for everything in that mixture, query it against the database, and get a list of species in a particular mixture. And that's going to be really important for monitoring environments. So for example, right now, we might look at sentinel species with things like malaise traps, where you would gather a bunch of insects, they hit the trap, climb up, and end up in the alcohol jar. There can be many different insect orders present in a single sample, and right now you'd need a legion of taxonomists to sort all of those specimens, try to identify them to species to the best of their ability. This simply doesn't scale. If we're trying to compare beta diversity across sites or understand our restoration effects over time, we need better tools that allow us to identify these kinds of bulk samples. And that's really the beauty of where this high throughput sequencing comes in, is that we can take that whole mix of bugs in the bottle, make it into a bug shake, and extract DNA from that slurry, do parallelized high throughput sequencing, and do that kind of species scan to get a list of what species are in that trap. So that's exciting because it's allowing us to move barcoding from single individuals or samples to metabarcoding, these biological communities, which is going to give us a much more integrative approach to this study of biological diversity at global scales. We're using these kinds of tools for environmental restoration at disturbed mine sites in northern Canada, trying to help understand how we can rehabilitate soils so that they support native plant communities. And we couldn't do this without this kind of metabarcoding approach. Organisms shed DNA into their environment. And this paper in 2008 was kind of a landmark study in showing that we didn't even need a specimen to do this kind of EDNA or detection of environmental DNA. So if you think about something like a frog or a fish, it's defecating into the water, shedding some epithelial cells. In the case of a fish, it's rinsing over the gills where epithelial shells are shed, the slime layer, gametes. So all of this material leaves residual DNA in the water that we can actually pull a water sample, filter it, extract DNA from the filter, and start to look at what species are in an aquatic system without ever having to even see the species. And that's an important development, because if we're worried about monitoring a population of species at risk, we don't want to be using traditional methods like electrofishing, gill netting, which are going to cause stress or even impart mortality on the very species we're looking to protect. So this ability to start to query species occurrence in the environment in a passive way is a pretty exciting development. And if you want to know a bit more about environmental DNA, there's a very nice research brief put together by the Great Lakes Fisheries Commission that has a number of information sheets and PowerPoint slides that you can share with students or managers that describe some of the uses and limitations of eDNA. So basically, I'll just quickly go over some of the highlights in that organisms shed DNA into the environment, and it can be detected using these traditional methods. Not all species are easy to catch using traditional methods, so this is also an advantage. Helps us to detect rare species, so even if you are doing electrofishing, you may not actually bump into that particular specimen in its environment. So this is a really powerful tool for applied biomonitoring. So we collect that water sample, isolate the DNA, extract it, and screen for its presence. And there are two pathways we can do that. We can look for a single species with qPCR, where we've made a species-specific primer and probe. So if we want to know, is red-side dace a species at risk in that creek, we can simply probe that DNA extract for red-side dace. And that's a very efficient way to analyze a lot of sites for a target species. Alternatively, we can use high-throughput sequencing to try and get an idea of the entire community in that water sample. So there are some things that can prevent eDNA from being detected. We know that high temperature and low pH can degrade eDNA, that some things like humic acid can act as inhibitors to PCR. So we need to take some measures to do workarounds and cleaning up extracts of inhibitors. But in general, it's a pretty powerful tool. Now, you can get a detection without, say, a live animal present. Could be contamination. This happens when people are moving their boats from one lake to another without cleaning them, or a moose moves from one wetland to another and is carrying eDNA on its pelt. Also, something like a dead fish may shed a lot of eDNA into the system because it's decomposing, and you might attribute that signal to, well, the fish is present, and technically it is, but it's dead. So there are some things that we have to think about in terms of interpreting that data. But what I find really exciting about environmental DNA is it allows us to interrogate a lot of different environments and at different timescales. So people have looked at metabarcoding eDNA from ice cores and can reconstruct what that environment looked like thousands of years ago based on the pollen grains and the DNA in them in these ice cores. They can look back hundreds of years by looking at soil horizons in a soil core or in sediments in a lake. Now, with aquatic systems, eDNA tends to degrade quite rapidly. So if we get an eDNA detection in an aquatic system, it's near contemporaneous with the occurrence of the organism. So we can begin to use that as to say, what's here right now? But if we really wanted to know what was in this lake in the past, we could look at a soil core of the sediment. So this is where we run into some of the limitations of eDNA in terms of just, we need better understanding of its origin. How much do different species shed? What state is it in? Is it clumps of cellular debris, or is it naked DNA suspended in the water? That has implications for what filter size you might use to capture it. Another question is, how far can eDNA be transported, say downstream, or how far does it diffuse from an animal, say a frog at the edge of a pond? And then what is the ultimate fate? How long does it persist in the system? So many academic studies are focusing around answering questions around the ecology of eDNA. And this is just a little conceptual diagram that shows how some of the factors can impact its degradation. There's a huge literature on this that's kind of outside the scope of today's discussion, but I just wanted to run through a couple of case studies, how we're using it. And this first use case was an on-site approach where we used a battery-operated pump in a backpack to essentially filter our water right on site rather than taking a water sample back to a lab. That's an advance because that eDNA can degrade in the water bottle from the time you take the sample at the site to getting it back to a lab. So that increases your risk of false negatives the longer that water sample sits in a jar. So in this case, we were just filtering the water directly out of the pond using an on-site extraction kit and a handheld PCR machine to detect the presence of Jefferson salamander, which is an endangered species here in Ontario, and it's part of a cryptic species complex together with the blue-spotted salamander, and it's very difficult, if not impossible, to tell apart morphologically. So we worked with some folks who had been studying Jefferson salamanders, and they knew that they were present in a number of pools. So we sampled eDNA from these 9 pools, analyzed them in triplicate. And what was interesting is that in all of the sites where there was known historical occurrence of the Jefferson salamander, we picked up eDNA from it as well. So it was a nice validation of the method. And its sensitivity was interesting because if you look at the second to the last entry on the bottom right, you'll see a yes for eDNA and a no for historical data. Because the Jefferson and blue spot salamander can live in mixed populations, they had missed it. They'd subsampled some number of animals and found that, well, they were blue spot, but there was, at a low frequency, a number of Jefferson animals in that site as well. So we also helped them recognize that one of the sites that they thought there were no Jefferson salamander, in fact, there were. Now, we wanted to also use this high throughput approach to doing a kind of metabarcoding of water samples. So we thought this would be an interesting validation to go to the Ripley's Aquarium in Toronto and sample at the Rainbow Reef, where they have more than 100 species of fishes in this aquarium. They knew the species composition of what was in there. We did this metabarcoding, and what we found was that using different markers, so you can see in the upper left of this figure, we used a 12S marker, a 16S marker, the cytochrome oxidase 1 marker, and because the PCR primers are not truly universal for any of those markers, we didn't get all of the species in the system, which was a bit depressing. Exciting because we still got a number of these animals with these methods, but we didn't get everything. So again, that's another limitation of this kind of eDNA metabarcoding, is some things may not even be in your reference library, so you get a sequence but no match. Or in other cases, you'll have sequences in the reference library, but the primers that you're using to amplify barcodes from that community sample aren't a good match for some of the species in that system. One of the big questions that people are very interested in, though, is asking, are number of sequence reads for a target species correlated with relative abundance in the system? And we could see general correlations that seem to match relative biomass of a species, but also a lot of outliers. Again, this seems to be from PCR amplification bias. Some things are overrepresented in terms of the number of sequence reads, others underrepresented. But in general, it looks like we can even start to get to relative abundance of different organisms in our systems using these methods. Another application where we used this eDNA metabarcoding was in looking at macrobenthic freshwater invertebrates. Now, macrobenthic freshwater inverts like caddisflies, stoneflies, mayflies are very sensitive to pollution. So they've traditionally been used as environmental indicators. And so traditionally, people would go out, look at a 1 meter quadrat, pull up all of the macrobenthic invert larvae they could find, bring them back to the lab and do morphological identification. We did that, but we also pulled an eDNA sample from the same site to see how well eDNA captured these macrobenthic inverts. And what really shocked us was that there was very little overlap. We were seeing quite a different community from the eDNA sample as what we saw from the morphological analysis of these specimens or a metabarcoding analysis of the bulk specimens. If we just did a find and grind of that, all of the specimens together, that gave us pretty similar results to the morphological study, but eDNA was quite different, which is interesting because we're starting to suspect that the macrobenthic invertebrate larvae don't shed as much eDNA. It probably settles at the sediment rather than getting suspended in the environment. So we have to use this tool cautiously. It's not going to immediately replace conventional surveys, unfortunately. So when we use these tools, we have to think a bit about this unholy trinity of the ecology of eDNA, the methodology of detection, and the environment that we're trying to operate in whether there may be PCR inhibitors or other things that would modulate our signal. But in general, I've been really impressed with the power of these tools and how they've been applied. I want to just come back for a minute to point out that DNA analysis is now being supported by a diverse array of platforms, from things like handheld qPCR machines to even handheld sequencers that can be plugged into a laptop in the field. So it's a very exciting time to be working on these platforms for molecular biodiversity research. And it's also important to note that we can take a different approach instead of just simply using a marker gene to identify what's in a mixture. We can do what they call deep sequencing. It doesn't rely on PCR. It's just sequencing all of the DNA in a sample. And that's an exciting development because we're not seeing PCR amplification bias interfering with the number of reads that we get. So we're getting a pretty good ability to start to quantify mixtures by doing this kind of deep sequencing without PCR. And of course, this kind of whole genome sequencing rather than marker gene sequencing is where the bacterial and microbial community has been for a long time. It's not enough to just identify something as anthrax. You might want to look for particular genetic inserts that would suggest that it's been weaponized. So microbial genomes are small. We're kind of moving beyond the barcode in those systems to just sequencing whole microbial genomes for sourcing things like antimicrobial resistance. And then I just want to finish up with my very last slide to talk about where I think the One Health initiative is going. This recent paper from Trends in Ecology and Evolution introduced the concept of a pathobiome. We have to get away from this idea of one organism causing disease, because we really actually are dealing with microbiomes of all sorts of things in the environment. So if you think about it, there's a fish gut microbiome, fish tissue microbiome, their water microbiome, the sediment microbiome, potential fish pathogens moving around in a system. And the point is that your severity of particular symptoms from, say, a fish pathogen may be modulated to a large extent by other players in the environment. So we need to move towards a functionally driven understanding of pathobiotic systems based on gene expression, metabolic interactions, and ecological processes. And that's where I think the whole field is going, and it's very exciting. And I'd just like to say thank you very much for your time.
Fantastic. I think we have time for one question. If someone wants to ask a question in the chat.
I think there's one there, Jamie.
Oh, perfect. The question is, with the COVID-19 virus, has there been a there's been a backlog in testing? Do you think that DNA barcoding could be applied to viruses? And if so, would we be able to use this as a more rapid form of testing to get results quicker from different samples like saliva?
Absolutely. In fact, we had a panel discussion in a course specifically dedicated to the pandemic a few weeks ago, where like microbes, viruses have very small genomes. In the case of SARS-CoV-2, the causative agent of COVID-19, they are sequencing the whole genomes to look at minor mutations as it transmits from one group to another. We can use that to kind of do a pathway analysis to say, is this the European strain of CoV-2 versus the Asian strain, and how are these things being introduced? But we can also look at closely related viruses and find viral specific gene regions that allow us to develop qPCR assays to do PCR on the RNA of, in this case, SARS-CoV-2 to do detection. And the qPCR tool is actually more sensitive than the antibody or antigen tests. So it's a very nice tool, but it does take a bit more time than say some of the antigen tests that are being developed. But we're already seeing a number of groups starting to commercialize these kinds of barcode-based assays for viruses, if you will.
Fantastic. I would like to thank our speaker for a fascinating and thought-provoking presentation. I really enjoyed the discussion, and I hope everyone did as well. Before we end, I want to remind everyone that the next One Health seminar will be on November 4th at 12:30, and our speaker will be Dr. Jennifer Provencher from the Environmental and Climate Change Canada, and the title of her presentation is Plastic Pollution and Wildlife Health. Thank you again for joining, and I hope to see you in 2 weeks.